Welcome to Monobiology. Today we're going to look at an introduction to the molecule, probably one of the most famous molecules, DNA. Now DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. And it is the molecule of heredity. We're going to talk about two things in this video. The first thing we're going to talk about is the structure of DNA, what it's made up of and how it's put together, as you can see here. We're also going to talk briefly about the function of DNA. Let's start. Before we can understand how DNA is put together, we have to understand the building blocks of DNA. Now remember that DNA stands for deoxyribonucleic acid. Okay, DNA stands for deoxyribonucleic acid. And its building blocks are as follows. They're called nucleotides. Okay, so DNA is made up of smaller individual Lego blocks, if you will, called nucleotides. You need a bunch of nucleotides to make up strands of DNA. And then you put the strands of DNA together and you get the actual structure of DNA. Now, what are the parts of this nucleotide? We have three parts. Number one, right here, we have what is called a nitrogenous base, literally spelled nitrogen, O-U-S, nitrogenous base. Okay, and there are four different ones, which we'll talk about in a second. And number two, we have a sugar. In this case, the sugar is called deoxyribose. And we have, for three, we have this area here, we have the phosphate group. Okay, so there's three parts. There's the nitrogenous base, there's the sugar, in this case deoxyribose, and a phosphate group. And those three things stuck together in this fashion compose a nucleotide. Now, there are, in fact, four different bases. These are the ones you've probably heard of before. The A's, the T's, the C's, and the G's. The names of those we'll get to in a second, but there are four different ones. So there's four different types of nucleotides, one with an A, one with a T, one with a C, one with a G. Let's now take these nucleotides and put them together to make a strand of DNA. To make a strand of DNA, you have to attach several nucleotides in a row. So here we have our original nucleotide, and next we have another nucleotide. It's going to go right here. Now how these attach together, so if you have two nucleotides about to form a strand of DNA, the phosphate group of one binds to the sugar group of the other, as you see here, right there. Okay, so let's get another one. Right here, put this guy here, connect them. And last but not least, we have one other guy here. Let's get him lined up properly, like that. Okay, so I want you to draw this. Make sure you draw this exactly as I've drawn it. Rather than drawing the structures, you've read the textbook, you see the different structures. We're not going to get into the different structures of these things. Okay, but we will get into using these shapes to draw them. Okay, so this is a single strand of DNA. Now, let's go through the names of the bases here. Okay, so this is adenine. Adenine. This one is guanine. This one is thymine. Remember, these are just the nitrogenous bases. And this one is cytosine. Okay, so those are your four bases. Now we have a single strand of DNA. Let's see how two strands of DNA come to make up the double-stranded DNA. All right, so to make our double-stranded DNA, we simply put our nucleotides together like so. Really easy, no problem. And we connect them all. Whoop, whoop, whoop. Remember, it's sugar to phosphate. Now, what we need to do next to build our double strand is we need to match up the complementary base pairs. We know that these are the bases A, G, T, and C, but they match up in particular with another base. A always matches up with T, and C always matches up with G, or vice versa. T always matches up with A, and G always matches up with C. So here we have an A, and we would like to actually match that up. What would we match it up with? It would be a T. So I'm going to find a T here, and I'm going to do this. Notice that I have placed the nucleotide upside down. Why did I do that? Because the strands are known as anti-parallel, which means that one is right side up and the other one is upside down. Okay, so here we have our right side up strand, and over here we're going to build an upside down strand. Okay, so anti-parallel. One's right side up and the other's upside down. So let's get our next nucleotide, and our next nucleotide to match up with the G here would be a C. I'm going to get that. I'm going to turn them upside down. Move them over a little bit. Here we go. And what matches up with T? That would be A. Get him. Put him right there. And remember, you're going to draw these two. The easiest way probably to draw these is to turn your paper upside down and to put these guys on. Kind of messed up the size a little bit here, but that's all right. Okay, and then what matches up with C? That would be G. Gonna turn him around and get him to look like that. Okay, don't forget when I do that, I'm going to connect my sugar to my phosphate, like so, and like so. Okay, the other thing you have to do is you have to connect your nitrogenous bases. We use dashed lines to represent weak interactions. Okay, so they're not strong chemical bonds. Notice that there are 2 between the T and the A, and there are 3 between the C and the G. I'm not doing a very good job of drawing these dotted lines, but you get the idea. Okay, so there's 3 between the C and the G and 2 between the T and the A. Okay, so there's our double strand of DNA. Anti-parallel, we've got one strand that's right side up and one strand that's upside down. They're connected in the middle here via hydrogen bonds. So these little bonds in here are H bonds. Those ones in there, and these ones down here. Okay, so the bonds between the nitrogenous bases are H bonds. Now, all that being said, you should be able to draw and label an anti-parallel double strand of DNA and label all the parts. Nucleotides, you should be able to identify the nitrogenous bases, the phosphate groups, the sugar, the H bonds, the strong chemical bonds. These ones here that are solid lines would be strong chemical bonds, etc. You should be able to do all of that. Now, when it's all said and done, you have your DNA double helix. Now I know that picture that we just looked at does not look like this. So what's going to happen in this particular situation is that there are going to be interactions among the different nucleotides that are going to coil the DNA so that it looks like a double helix, and that's what it's called. So a helix would be like a spring. This is like a double spring. Okay, so it's a spring attached to another spring and coiled all around itself. And that's what we have. We have the double-stranded DNA. Now, the functions, very quickly. DNA, very simply put, is the code for all of the proteins that exist within our bodies. It houses the blueprints for all of the proteins in our body. The proteins do all of the work of the cell. Very important. That in a nutshell is DNA. Intro to DNA. Bring your questions to class.